Our Chapter 34 lecture covers the topic of animal nervous systems. Let's begin by taking a look at this interaction between a cheetah and its gazelle prey. This cheetah detects a herd of grazing gazelles with its keen vision. The cheetah's ability to recognize wind direction enables it to move downwind of the gazelles to minimize auditory and olfactory cues that might otherwise be carried by the wind toward the suspecting prey. Olfactory and auditory cues warn the gazelles of a cheetah's approach. When does the cheetah decide to sprint in pursuit of the prey and the gazelle to turn and run? The perception and the reactions of these two animals depends on the activity of their nervous systems. The nervous system takes in information from the environment through the animal's sensory organs and processes that information through the activity of networks of nerve cells. The results of the processing guide an animal's motor responses, such as the flight of a gazelle and its pursuit by a cheetah. As we introduced in the last chapter, a nervous system is fundamental to maintaining homeostasis in the vast majority of animals. Except for sponges, all multicellular animals possess a nervous system, a network of many interconnected nerve cells. This network allows an animal to sense and respond to the environment, coordinate movement through the action of muscles, and regulate the internal function of its body. The basic functional unit of the nervous system is the neuron. Most nerve cells have dendrites, which are fiber-like extensions that receive information, and axons, other extensions that transmit information. Rapid communication between neurons is achieved via the electrical properties of the neurons. All animal nervous systems are made up of three types of nerve cells. Sensory cells send and receive information about an animal's environment or its internal physiological state. They respond to physical features such as temperature, light, and touch, or to chemical signals such as odor and taste. Interneurons process the information received by sensory neurons and transmit it to different body regions, communicating with motor neurons at the end of a pathway. Motor neurons produce the suitable organismal responses. The result is a network of interconnected nerve cells that form a circuit. These circuits allow information to be received, processed, and delivered. In most animals, interneurons form specialized circuits that may consist of a diverse array of nerve cells having different shapes, sizes, and chemical properties. Nervous systems can range from simple to complex. The only multicellular organism to lack a nervous system is the sponges. Their body plan is so simple that a nervous system is not required to coordinate the functions of different body regions. Instead, groups of cells within sponges respond to local chemical and physical cues. Nervous systems first evolved in simple animals. The simplest nervous systems can be found in the cnidarians, which include jellyfish and sea anemones. These nervous systems have relatively few neurons. The nervous cells of these animals are arranged like a net. More complex systems can be found in the bilaterians, which make up the rest of the animals and are defined by the characteristic of bilateral symmetry. The early nervous systems could sense basic features of the environment, such as light, temperature, chemical odors, and physical forces. Guided by these cues, early nervous systems could trigger movements useful to obtaining food, finding a mate, or choosing a suitable habitat. As animals evolved more complex bodies with increasingly specialized organ systems for respiration, circulation, digestion, and reproduction, their nervous systems also became more complex. The ability to process information first evolved with the formation of ganglia. Ganglia are groups of nerve cell bodies that process sensory information received from a local nearby region. Ganglia send a signal to motor neurons that control some physiological function of the animal. They are the processing points in nerve cell circuits. In animals with a highly organized nervous system, information is transmitted to different regions along the length of the animal's body by distinct nerve cords and nerves. These are bundles of long, fiber-like extensions from multiple nerve cells. In earthworms, paired ganglia are located in each of the body's segments. Each ganglion is made up of many interneurons. These segmental ganglia help to regulate the muscles within each body segment that moves the earthworm's body. Eventually, the centralized collections of neurons forming a brain evolved. The brain was able to process increasingly complex sensory stimuli received from the environment or body. Centralized collections of neurons forming a brain are present in annelid worms, insects, and mollusks. Sensory organs also developed in the head region. These ranged from eye spots in flatworms that respond to light to more sophisticated image-forming eyes in octopus and squid. The evolution of a brain enabled these invertebrate animals to learn and perform complex behaviors. The evolution of the brain is also a hallmark of vertebrate animals. It enabled them to evolve complex behaviors that rely on learning and memory, and, in certain vertebrates, the ability to reason. At the same time, animal brains became increasingly complex in the number of nerve cells they contain, in the number of connections between neurons, and in the variety of nerve cell features. As a result of natural selection for enhanced sensory perception and the ability to respond to cues in the environment, the nervous systems of many animals are complex. Their complexity is linked to more sophisticated decision-making abilities and behaviors. Neurons all have some basic features in common. First, they have a large cell body. Second, they have dendrites that act as inputs for the nerve cell. And third, they have axons that serve as outputs for the cell. Both types of cellular extensions, dendrites and axons, can be highly branched. The branching enables neurons to communicate over large distance with many other cells. The end of each axon forms a swelling called the axon terminal. An axon terminal communicates with a neighboring cell through a junction called a synapse. A space called the synaptic cleft separates the end of the axon of the presynaptic cell and the neighboring postsynaptic cell. The synaptic cleft is commonly only about 10 to 20 nanometers wide. All nerve cells use fundamentally the same mechanisms to communicate with neighboring cells. First, a neuron's dendrites receive signals from other nerve cells, or, in the case of sensory nerves, from specialized sensory endings. These signals travel along the dendrites to the neuron's cell body. Second, the signals are summed up at a region called the axon hillock, where the cell body and its axon join. Next, if the sum of the signals is high enough, the neuron fires an action potential. An action potential is a brief electric signal transmitted from the cell body along one or more axon branches. The arrival of a nerve signal at the axon terminal triggers the release of neurotransmitter molecules from vesicles. The neurotransmitters convey the signal from the end of the axon to the postsynaptic target cell. The molecules diffuse across the synaptic cleft and bind to receptors on the plasma membrane of the target cell. This binding causes a change in the electrical charge across the membrane of the receiving postsynaptic cell, continuing the signal to the second cell. Most neurons form hundreds to thousands of synapses with other cells. The massive number of interconnections enables the formation of complex nerve cell circuits and the transmission of information from one circuit to others in the nervous system. It's important to recognize that some neurons do not synapse with other neurons. Instead, they synapse with other cell types that produce a response. For example, muscle cells or secretory cells. Neurons show an amazing diversity in size and shape. They can differ in terms of length, the position of the cell body, and/or the number of extensions coming from the cell body. Some neurons, such as interneurons, are very short. In contrast, the motor neuron that extends from your spinal cord to your big toe is several feet long. Most of that length is in the axon. A neuron's dendrites form a finely branched field that receives inputs from many other nerve cells or receptors. The degree of branching can vary among neurons. A neuron with more dendritic branches receives input from more sources than one with fewer branches. All neurons are electrically excitable cells. Nerve signals travel at high speeds ranging up to 200 meters per second, or approximately 450 miles per hour. The speed of nerve signals is fast by biological standards, but low by comparison with the speed of electrical signals transmitted by a wire. The inside surface of a neuron carries an electrical charge that is due to the presence of charged ions, and the same is true of the outside surface. The electronic charge on the inside and the electronic charge on the outside are not the same because unequal numbers of charged sodium and potassium ions are located inside and outside the cell. Potassium ions are identified by their chemical shorthand of K+, and sodium ions are designated by their chemical shorthand of Na+. The charge difference between the inside and the outside of the cell due to differences in charged ions is the neuron's membrane potential. This difference in electrical charge is measured in volts. Other cells have membrane potentials, but only muscle cells and nerve cells respond to changes in their membrane potential. At rest, when no signal is being received or sent, the cell's membrane voltage is negative on its inside relative to its outside. This is shown here in the voltage graph on the right, a condition in which the cell is said to be polarized. This means there is a buildup of negatively charged ions on the inside surface of the cell's plasma membrane and positively charged ions on its outer surface. The negative voltage across the membrane at rest is referred to as the cell's resting membrane potential. The resting membrane potential ranges from -40 to -85 millivolts, depending on the type of nerve cell. The sodium-potassium pump, shown here as the green proteins, uses the energy of ATP to move 3 sodium ions outside the cell for every 2 potassium ions moved in. But potassium channels in the cell membrane, shown here in pink, allow some potassium ions to leak out of the cell. This steady loss of positive charge from inside the cell makes the inside of a resting nerve cell more negative than the outside of the cell. This is what determines the resting membrane potential. Before we dig into the details of how an action potential is generated, propagated down the length of the neuron, and transferred to a following neuron, I want to note that these functions are dynamic processes that involve chemical movements into and out of a neuron, as well as chemical transmission between neurons. Therefore, I want to draw your attention to media available in Chapter 34 on the How Life Works Launchpad site. These animations include how action potentials are generated, how these signals are propagated, and then how signals are transferred to other neurons at chemical synapses. Action potentials serve as the primary mode of communication down the length of a neuron. This process is electrochemical in the sense that the relative movement of charged ions leads to a change in the electrical gradient formed between the inside and outside of the neuron cell. The first step toward the process of creating the action potential signal involves receiving a signal at the dendrites. Starting at the terminal end of the dendrite, in response to neurotransmitter binding to membrane receptors, a small electrical signal travels to the cell body. At the axon hillock, the summed membrane depolarization of the cell's dendrites causes voltage-gated sodium channels to open, allowing sodium ions to enter the cell. This kicks off the full automatic depolarization of the neuron action potential. When a nerve cell is excited, its membrane potential becomes less negative, meaning that the inside of the cell becomes less negative or more positive than the outside of the cell. The increase in membrane potential is referred to as depolarization of the membrane. If the excitatory signal is strong enough to depolarize the membrane of the nerve cell body to a voltage of approximately 15 millivolts above the resting membrane potential, the nerve fires an action potential at the axon hillock. An action potential is a rapid, short-lasting rise and fall in membrane potential. The critical depolarization voltage of 50 millivolts required for an action potential is the cell's threshold potential. A nerve cell fires an action potential in an all-or-nothing fashion. The first step in a firing of an action potential involves the cell membrane crossing its potential threshold at the axon hillock. Next, a large number of voltage-gated sodium channels open, allowing sodium ions to rush inside the cell. The rise in voltage that results causes additional voltage-gated sodium channels to open. This is an example of positive feedback. A signal, in this case depolarization, causes a response, which is the open voltage-gated sodium channels, and this leads to an enhancement of the signal that leads to an even larger response. The rising phase of the action potential is followed by the falling phase, repolarization. This sudden reversal is caused by two important factors. One is voltage-gated sodium channels closing. The second is voltage-gated potassium channels opening. As a result of the closing of voltage-gated sodium channels and the opening of voltage-gated potassium channels, the membrane voltage peaks and then rapidly falls as potassium ions move out of the axon through open voltage-gated potassium ion channels. The voltage briefly falls below the resting membrane potential. The voltage returns to the resting potential after another few milliseconds as potassium ion channels close and restore the resting concentration of sodium and potassium ions on either side of the cell membrane. The period during which the inner membrane voltage falls below and then returns to the resting potential is called the refractory period. During this time, a neuron cannot fire a second action potential. Part of the reason is that when voltage-gated sodium channels close, they require a certain amount of time before they will open again in response to a new wave of depolarization. The duration of the refractory period varies for different kinds of nerve cells, but limits a nerve cell's fastest firing frequency to less than 200 times per second. Most neurons code information by changing the rate and timing of action potentials. Generally, a higher firing frequency codes for a more intense stimulus, such as a brighter light or a louder noise, or a stronger signal transmitted by the nerve cell to other cells it contacts. Once initiated, an action potential propagates along the axon. The local membrane depolarization initiated at the axon hillock triggers the opening of neighboring voltage-gated sodium channels further along the axon. The inward sodium current depolarizes the membrane above threshold, triggering the opening of nearby voltage-gated sodium channels still further along the axon. The depolarization spreads down the axon in this manner. Neighboring voltage-gated potassium channels subsequently also open and close to reestablish a resting membrane potential after an action potential has fired. Action potentials propagate only in one direction, generally from cell body at the axon hillock to the axon terminal. The refractory period prevents the membrane from reaching threshold and firing an action potential in the reverse direction. Neurons are supported by other types of cells that do not themselves transmit electrical signals. One such type of cell is the glial cell. The human brain has many more glial cells than neurons. Glial cells surround neurons and provide them with nutrition and physical support. Since nerve cells have a limited capacity to regenerate after being damaged, protection of the brain and spinal cord is critically important. During development, glial cells help orient neurons as they develop their connections. Glial cells also provide electrical insulation to vertebrate neurons. Glial cells form multiple lipid-rich layers or sheaths called myelin that wrap around the axons of neurons. Myelin gives many nerves their glistening white appearance. By insulating the axon, myelin greatly increases the speed of nerve signal transmission, which allows the nervous system to respond rapidly to stimuli. Glial cells can be further distinguished based on a variety of functional roles. Glial cells called Schwann cells insulate sensory and motor neurons. Glial cells called oligodendrocytes insulate cells in the brain and spinal cord. And star-shaped glial cells, called astrocytes, contribute to the blood-brain barrier. This set of structural adaptations affects the blood vessels supplying the brain, and it prevents pathogens and toxic compounds in the blood from entering the brain. The conduction speed of action potentials is limited by neurons' membrane properties and the size of its axon. The myelin sheath insulates the axon's membrane, spreading the charge from a local action potential over a much greater distance on the axon's length than would be possible in the absence of myelin. At regular intervals, the axon membrane is exposed at sites called nodes of Ranvier. Voltage-gated sodium and potassium channels are concentrated at these nodes. As a result, the action potential in myelinated axons jumps from node to node. Saltatory propagation greatly increases the speed of signal transmission, enabling vertebrate animals to respond rapidly to stimuli in their environment. How did science first learn about resting and action potentials? The action potential was first measured in 1939 by two British neurophysiologists, Alan Hodgkin and Andrew Huxley. They inserted a small electrical recording device called a microelectrode into a squid neuron to record the voltage of the inside of a nerve cell relative to the outside. Squid neurons are a great model system because the relatively large size of their nerve cells makes them easier to study. Hodgkins and Huxley then used a separate set of electrodes to depolarize the cell to threshold, triggering an action potential. The graph on the right shows the resting membrane potential and the course of an action potential recorded by the electrode. Subsequent work focused on identifying the ion channels responsible for the changes in current across the membrane. As we've previously discussed, neurons communicate at specialized junctions called synapses. The two types of synapses are electrical and chemical. Electrical synapses provide direct electrical communication through gap junctions that form between neighboring cells. These enable rapid communication but limit the ability to process and integrate information. Chemical synapses are the more common type of synapse and are shown here. The signals conveyed at chemical synapses are chemicals called neurotransmitters. These molecules are contained within small vesicles in the knob-like axon terminal. The steps in synaptic transmission begin with an action potential reaching the end of an axon. The depolarization of the action potential induces voltage-gated calcium ion channels in the axon terminal membrane to open up. Because the concentration of calcium ions, denoted here as Ca2+, is higher outside the cell, calcium ions diffuse through these channels into the axon terminal. In response to the rise in calcium concentration, the vesicles fuse with the presynaptic membrane and release neurotransmitter molecules into the synaptic cleft by exocytosis. The neurotransmitters diffuse rapidly across the cleft and bind to postsynaptic membrane receptors of the neighboring cell. The binding of neurotransmitters causes a change in the postsynaptic cell membrane potential, allowing the signal to propagate along the next neuron through action potentials. The neurotransmitter's effect on the postsynaptic membrane ends shortly after binding. Neurotransmitters become unbound from the receptor. Neurotransmitters in the synaptic cleft are either broken down by a deactivating enzyme or taken up again by the presynaptic cells and reassembled in newly formed vesicles. Signals between neurons can be excitatory or inhibitory. The binding of some neurotransmitters depolarizes the postsynaptic membrane, making it more positive. The positive change in membrane potential is termed an excitatory postsynaptic potential, or EPSP. The binding of other neurotransmitters can hyperpolarize the postsynaptic membrane, making it more negative. In this case, the negative change in membrane potential is called an inhibitory postsynaptic potential, or IPSP. Excitatory synapses tend to transmit relative information between nerve cells. Inhibitory synapses, in contrast, often serve to filter out unimportant information. For example, when a grazing gazelle senses a predator, its nervous system must transmit the key sensory information about the presence of a predator, but filter out irrelevant information such as the flies buzzing over its back and ears. Inhibitory synapses are also important for controlling the timing of muscle activity required for coordinated movement. The postsynaptic nerve cell sums the excitatory and inhibitory synaptic inputs that it receives through its dendrites and cell body. If the sum of these synaptic inputs results in a membrane potential that exceeds threshold at the axon hillock, the neuron fires an action potential. If the sum of inputs does not exceed threshold, no action potential fires. EPSPs and IPSPs can be summed over both time and space. When summed over time, temporal summation, the frequency of synaptic stimuli determines whether the postsynaptic cells fires an action potential. When summed over space, the number of synaptic stimuli received from different regions of the postsynaptic cell's dendrites determines if the cell fires an action potential. Sometimes excitatory and inhibitory signals may cancel each other out. Temporal and spatial summation of EPSPs and IPSPs are the fundamental forms of information processing carried out by the nerve system. Now that we better understand individual nerve function, how does the totality of nerve activity coordinate within animal bodies? The body's command center is the brain. It receives sensory information from the eyes, ears, nose, tongue, skin, and internal organs, and sends instructions to the rest of the body. As animals evolved the ability to sense and coordinate responses to increasingly complex stimuli, their nerve systems became organized into peripheral and central components. Nerves form the line of communications between these nervous system structures. Nerve cell bodies are grouped compactly together in sensory organs, in ganglia, and within a main nerve cord that extends from the brain. Nerves are composed mainly of axons from many different nerve cells. The peripheral nervous system, or PNS, shown here in blue, is comprised of both sensory and motor nerves. The anatomy of the peripheral nerve system is organized into two areas. The left and right side of cranial nerves are located within the head. Cranial nerves link specialized sensory organs, including the eyes, ears, and tongue, to the brain and control eye movement, facial expressions, speech, and feeding. The set of spinal nerves runs from the spinal cord to the periphery of the body. Spinal nerves exit from the spinal cord through openings located between adjacent vertebrae to thread through the trunk and limbs of an animal's body. Spinal nerves receive sensory information from receptors in nearby body regions along the length of the body. Spinal nerves also send motor signals from the spinal cord back to those sensory regions. The peripheral nervous system also includes interneurons and ganglia that integrate and process information in local regions of the animal's body. In contrast, the central nervous system, or CNS, encompasses the brain and centralized information ganglia. These are shown here as the yellow neurons. The bulk of information processing, particularly in animals that exhibit a great capacity for learning and memory, occurs in the central nervous system. The CNS of vertebrates includes both the brain and the spinal cord. The spinal cord is a central tract of neurons that pass through the vertebrae to transmit information between the brain and the periphery of the body. Nerves communicate between these two parts of the nervous system. Sensory organs at or near the body's surface transmit information by afferent neurons, which send information toward the central nervous system. After processing the information, the ganglia and brain send signals through efferent neurons, which send information away from the central nervous system. These commands coordinate the activity of muscles and glands in different regions of the animal's body. This organization ensures that an animal achieves a coordinated behavioral and physiological response to the stimuli received from its environment. The peripheral nervous system has voluntary and involuntary components. When a gazelle senses a predator, some nerve circuits send a signal to run, an action that is under conscious control. Other nerve circuits signal the heart to beat faster and blood vessels supplying muscles to dilate, actions that occur unconsciously. Conscious reactions are under the control of the voluntary component of the nervous system, and unconscious ones are under the control of the involuntary component. Voluntary components mainly handle sensing and responding to external stimuli. Involuntary components typically regulate internal bodily functions. In vertebrates, the peripheral nervous system is divided into somatic, or voluntary, and autonomic, or involuntary, components. The somatic nervous system is made up of sensory neurons that respond to external stimuli and motor neurons that synapse with voluntary muscles. This system is considered voluntary because it is under conscious control. However, many reflexes are controlled by the brainstem, independent of conscious control by the nervous system. The autonomic nervous system controls internal functions of the body, such as heart rate, blood flow, digestion, excretion, and temperature. It includes both sensory and motor components, which usually act without our conscious awareness. The autonomic nervous system is divided into two major subdivisions, sympathetic and parasympathetic. Both continuously monitor and regulate internal function of the body. Generally, sympathetic and parasympathetic nerves have opposite effects, but not always. For example, sympathetic neurons stimulate the heart to beat faster, whereas parasympathetic neurons cause the heart to beat slower. Reflexes, despite their seemingly automatic nature, fall under the category of somatic voluntary control. Simple reflex circuits make fast responses possible. Reflex circuits bypass the brain by directly connecting sensory neurons with motor neurons. Reflex circuits are common in both the somatic and autonomic components of the nervous system. The knee-jerk reflex in humans is an example of a simple nerve circuit. This circuit includes only a single synapse between two neurons, a sensory neuron and a motor neuron. Physicians commonly use this reflex to evaluate peripheral nervous and muscular system function. First, the physician provides a stimulus to the patellar tendon. Next, a signal from the muscle stretch receptor signals a motor neuron in the spinal cord. This leads to increasing the activation of the extensor thigh muscle, causing the muscle to contract and the leg to extend at the knee. Because muscles can only contract, opposing movements at a joint such as flexion and extension require flexor and extensor muscles on either side of the joint. Often, one set of muscles is activated and the other is inhibited. This pattern of joint and limb movement is achieved by reciprocal inhibition. When stretch receptors of the knee extensor are activated, they also inhibit the activity of opposing muscles that flex the knee. Reciprocal inhibition helps to control the movement of the right and left limbs during locomotion. This concludes our discussion of the general function of the nervous system. While we do not specifically cover sensory organs in this course, I encourage you to continue reading the remainder of Chapter 34 to learn more about these specialized senses.